from Oxford University, and he'll talk about improving scoring functions for protein binding, protein-ligand binding.
I don't use Max. Here, you'll be faster.
The next speaker, please set up early enough that we don't lose time. It's Altner and Zhangxing.
Okay. There you go. Cheers. Someone who uses Macs, tell me how to go presentation mode on a Mac, please. Should have just used my laptop after all.
You cannot do bioinformatics and use Windows, I'm sorry.
I use Linux, come on.
Okay.
That's where it's at.
Then you're okay. What is it? Slideshow?
There we go. Right, brilliant. Okay, so sorry about the technical difficulties. I'll try and make up time as we go along. Oh, bloody hell. This is also very sensitive. Okay, hi. So first off, I'd just like to thank the ISMB organizers for inviting me to present my work today. So I am the second Fergus from Oxford who's going to be speaking today, and I am also going to be talking about something related to small molecules, so sorry. This is some work that I've done over the last year with Charlotte Dean and Garrett Morris, where we focused on using descriptors of the small molecule ligand to help improve the performance of scoring functions for protein-ligand binding affinity. Oh god, what's happened now? Okay, we're good? Cool. Right, so many of the talks today have mentioned the use of scoring functions in various contexts. I imagine most people are familiar with the concept of scoring functions or energy functions in one context or another, be it protein-ligand docking and binding, protein-protein interactions, or protein structure prediction, or indeed anything else. But just to motivate why we care about scoring functions for protein-ligand binding affinity, so first off, this is still very much an unsolved problem. So-called classical scoring functions, which are used by your favorite protein-ligand docking software, be that Glide, Autodock, whatever, tend to be reasonably successful at identifying near-native binding modes and ranking active compounds over inactive, but they still remain relatively poor at actually estimating the binding affinity of a given ligand for a protein target. And although there is a lot of literature in the recent years which have shown that various machine learning approaches to this problem are able to consistently outperform these classical methods, there's still some way to go. And so the reason we care about this is that accurate scoring functions for protein-ligand binding affinity are important in early-stage drug discovery, because you want to be able to quickly and efficiently filter very large compound libraries against a target of interest. And also, from a scientific perspective, if you know that you've developed a good model that is able to accurately estimate protein-ligand binding affinity given a single snapshot of what is fundamentally a dynamic biophysical system, analyzing that model might be able to tell you a little bit more about what's actually going on in terms of molecular recognition. So that's quite interesting. So most methods, be they the simple classical methods or more recently the machine learning methods, focus on utilizing a 3D structure of the protein-ligand complex, specifically characterizing interactions between the protein and the ligand. Very little information about the ligand itself is directly used. So the question we asked here is would that information be useful? Would a more detailed chemical description of the ligand be helpful for predicting binding affinity? So just to, for those who aren't familiar with binding affinity prediction, just to give an idea of the sort of methods that are normally used. When I say scoring functions are typically structure-based, what I mean is you have a 3D structure of the protein-ligand complex of interest. Typically, this would be a docked pose. This is just a crystal pose from the PDB. And the sort of things that you might try to use to characterize that are, some people have mentioned previously, the concept of protein-ligand interaction fingerprints. You might use force field terms, or you might use descriptors capturing potential interactions. What these all have in common is they're pre-computed features that you generate from a 3D structure. And again, historically, you would tend to use these as features in a linear regression model. More recently, everyone's applied their favorite machine learning algorithm using engineered features and shown that it works a little bit better. In contrast, when I say ligand-based feature, I mean a feature which is computed solely from the ligand itself. And in particular, everything I'm going to focus on today is entirely based on the chemical properties of the ligand, i.e. independent of its 3D conformation. So you don't need an accurate docked pose of the ligand to compute these things. So the sort of things that we're working with, in this case, we just computed a set of molecular descriptors implemented in RDKit. There's about 180 of them we worked with. And the sort of things that you might compute about a ligand are physical and chemical properties. For example, some easily interpretable ones are its molecular weight, how many hydrogen bond donors or acceptors does it have, measures of properties such as polarizability, which you can capture through things like molar refractivity, or lipophilicity, which is captured by computed log P. And so the reason you might think this information might be useful is that these sorts of features have been successfully used in building models in QSAR, where so you have a target of interest, and you want to model the activity of your set of compounds. But a more detailed set of features such as this still is used in relatively limited ways in universal scoring function development. And just to, I mentioned that machine learning methods have been shown to consistently outperform so-called classical methods, but that's just an empirical observation. Why, other than the gain in performance, why might it actually be beneficial in general to use machine learning? Or to put it another way, why do we think machine learning might be doing better? So the first thing to stress is that unless you're working with multitask neural networks, for example, you typically need to train for a given task. So for example, if you want to train for if you want to do pose prediction, you need to train for that. If you want to do affinity prediction, like we're doing here, you need to train specifically for that. And chances are one model won't do well at both. But the advantages of doing this are that we see how many more structures are deposited in the PDB every year. We see how much affinity data is deposited in Chembl every year. Linear models can't really learn from all of this data, but we'd really like to do that. And common to most machine learning methods is the ability to scale with large amounts of data and continue to learn from that data as long as it's relevant. And also, another subtlety of this is that when you use a linear method, or indeed any other such method where you're imposing a functional form, you make an assumption about, I have this feature, I expect it to contribute to the affinity in this manner, be it in a linear or a nonlinear way. Instead, if you use your favorite machine learning method, you're able to directly infer those relationships from the data without imposing a functional form. And just as an aside, we tested several machine learning methods. We found the ensemble methods, and in particular random forest, consistently performed best. So what did we do? We did what everyone in scoring function development does, so we set our sights on validating using the test sets from the comparative assessment of scoring function exercises, CAS-F. First one was published in 2009, then 2013, then 2016. These are just sets of, at first 200, and then going up to 280 structures from the PDB with a measured binding affinity. And they're all taken from the PDB bind database. So we set those as our validation sets. And then for training data, we took everything in the most recent release of the PDB bind general set. And so the naive thing you would do is just train on all that data, test on those validation sets, and say, oh, hey, look, I did really well. My number's bigger than yours. Which we did. I'll show you that in a minute. But one of the other things that we wanted to look at is, by construction, there's a lot of similarity between the proteins in that training and test set. So we wanted to investigate how does that bias affect the performance of the models. So we then repeated the training process by excluding proteins from the training set if the protein was above a certain sequence identity percent to any of the proteins in any of the validation sets. And we varied the cutoff of sequence identity at which we did this. So first off, my number's bigger than your number. For two approaches we tested, we took the scoring function used by Autodoc Wiener and RFscore version 3. It's one of the state-of-the-art ML methods out in the literature. We took the features used by these two scoring functions, both structure-based, and we retrained a random forest using those features, and then trained a second random forest using both those features and the molecular descriptors computed using RDKit. And what we found is, so in the first 4 rows, you have the performance of the method without and with the RDKit molecular descriptors, and consistently the method using the RDKit descriptors performs better, be it on cross-validation, any of the CAS-F sets, or on the combination of CAS-F structures. RScore version 3 doesn't gain much performance on CAS-F 2016, but it already did quite well there anyway, so I suspect that's probably why. We also looked at what happens when you use only the RDKit molecular descriptors. Only the information about the ligand, the 2D information. You don't even have the conformer of the ligand in there. And what we found was that actually a random forest using those features gets a pretty damn high correlation coefficient on all of the test sets. For reference, your favorite classical scoring function is hovering around Pearson correlation of 0.6, maybe 6.5, depending on how much you care about significant figures. So this shouldn't be doing that well. Why is it doing that well? We'll get onto that in a minute. And just very briefly, I'm now going to focus purely on R-score version 3, but we observed the same results with several other scoring functions. You can talk to me in more detail afterwards if you like. And here I've plotted the correlation coefficient on the combined CAS-F set, just all structures from all CAS-F exercises, when you train the model only on data for which the training proteins are below a certain sequence identity to anything in the test set. The rightmost point, where you have no cutoff, corresponds to the data on the previous slide. And then as you move from right to left, you gradually impose a more stringent sequence identity cutoff. And we observe a couple of interesting things. The first one is that, unsurprisingly, performance gets worse as you drop off. But what's interesting is we controlled for this by also varying the size of the training set. Chronologically, in this case, you can do it other ways. And that didn't have much of an impact. So the drop-off in performance here can be attributed entirely to the removal of the most relevant data, not just removing data. Secondly, the red line, corresponding to the model augmented with the ligand-based features, is consistently above the blue line, the model not augmented. And again, it was the same regardless of which structure-based method we used. I'm just showing you one here because the other plot looks ugly. The dotted line is the model using only the RD-kit descriptors. Notice how that dotted line remains above 0.6 until you get down to removing lots and lots of data. Even when you remove nearly identical stuff, and similar ligand tends to correspond to similar protein, so it's unsurprising that this model is affected by this, until you get down to that really stringent cutoff, that quote-unquote ligand-based scoring function is still achieving a comparable result on this benchmark to your favorite classical scoring function. So what's going on? Well, firstly, we just looked at what is the random forest actually picking out as useful features from the ligand. And it's things like molar refractivity, van der Waals surface area contributions, computed log P, topological polar surface area, et cetera, et cetera. We verified there was no trivial correlation between any of these features and binding affinity, as you do. So whatever relationship is being inferred is more complex than just a trivial bias in the data. You can try and rationalize why some of these features might be useful. For example, molar refractivity tells you about polarizability. Log P tells you about lipophilicity. This might tell you something about the ligand's propensity to sit in a certain type of binding pocket. But you would still expect to need the binding pocket to actually use that information. So while this might tell us a little about why these features are useful, we still don't fully know why this model is doing so well on this benchmark. This raises questions about benchmarking, but I'm not going into that here. And it was the same set of features that were useful when you combined them with any set of structure-based features that we tried. So it wasn't a quirk of one model. So I'm just going to very quickly try and address the question of what the hell is actually going on when we're using this purely ligand-based model. And to do this, what we did is we looked in the PDB-Bind database, and we identified groups of structures which feature the same ligand. For example, there were on the order of about 20 structures binding biotin. So you had avidin, streptavidin, and a bunch of other engineered proteins. And for all these cases where you had multiple ligands, the same ligand in multiple structures, we took that group of data, we trained a random forest using only the ligand-based features on the remaining data, we made the singular prediction that that model would make for just that ligand, and we plotted it against the spread of experimental data and computed the correlation between some summary statistics. What I have here are swarm plots of the experimental data for some of the most common ligands. Biotin's an obvious outlier here, right in the middle, crazy high affinity. But for everything else, the red marker corresponds to what the ligand-based method is doing, and when you aggregate this over all of the data, you actually have quite a strong correlation between the predicted and mean affinity. So however it's doing it, the inclusion of the ligand-based features in the model appears to be estimating the mean affinity of that ligand for its observed binding partners, at least empirically in this data set. So the really obvious thing here is, well, if you have a structure-based method that can tell the difference between different targets and different poses to the same target, it should be able to more accurately reproduce that spread of experimental data. So we repeated exactly the same experiment using R-score version 3, trained it on everything but that ligand, tested it on structures with that ligand, and we found that, so the red markers show the ligand-based only predictions again. The swarm plots here show the predictions that R-score version 3 is making for each of the structures featuring a particular ligand. So in the leftmost swarm, for example, everything binding ADP. And what we found is that not only, if I just flick back to the previous slide, not only is the model failing to predict the range of experimental data for any of the ligands here. There were more ligands, it's the same even in the ones not shown here. But also, in most cases, it's not even predicting them in the right order. So, for example, for ADP, there was no correlation essentially between the predicted affinity and the experimental affinity. Sometimes there was, sometimes there wasn't. Usually there wasn't. When there was, half of the time it was 2 data points, so statistical significance is not there. But I believe that still needs more work to understand what's fully going on. But just to conclude, it appears to be the case that ligand-based features consistently improve scoring functions for binding affinity. And we also observed strong homology bias between training and validation structures and how this affects scoring function performance. We looked into the ligand-based features, found they appear to be predictive of mean affinity. And we also found while doing this that some of the structure-based methods we were using completely failed to predict the actual experimental data for ligands against multiple targets. I'd just like to acknowledge my supervisors, Garrett Morris and Charlotte Dean. Thank you all for listening. Got a poster downstairs, A32. It's a preprint on archive. All the materials to reproduce this is on our website. Thank you.
Okay, are there questions?
Hi. Thanks for the talk. Very nice methodology. I'm wondering whether you consider to apply to the D3R challenge to see where you stand. So it's a similar challenge as Capri, CASP, so it's for...
Yeah, no, so definitely when I was working on this, calls were already out for people to submit to D3R at the time. Certainly one of the things that I want to look at is testing, not to claim we did better or worse than anyone else, but I'd be interested to see how all of these different methods perform on an external prospective validation set. In particular, if you vary the training set construction given what we know, how does that affect something completely external? Yeah.
Okay. Thank you for the nice talk. I just wanted to know if you considered using some protein features, like features from the binding site of the protein to increase the accuracy.
Yeah, it's a very good question. One of the obvious things to do. So I didn't do that for this work. Obviously, things you could try and evaluate are, for example, depth of the binding site, or even what residues are in there that could potentially form certain interactions. So I haven't looked at that yet. There's some literature on this. I don't think that's been fully explored either. But it would definitely be interesting to see how that, especially in concert with the ligand-based features, how useful that might be.
Okay. Second question. You also used a different version of the CAS database. I think they are quite redundant. I'm not sure if there is any significant difference between the different data sets that you use for validation.
So the CASF validation sets?
Yeah.
Yeah, so there is... So there's some overlap between the 2013 and 2016 CASF sets, so they're the PDB-bind core sets. About 50% of the structures in the 2016 core set are also in the 2013 core set. The authors of CASF themselves suggest that you use both because they're different, or there's some difference in there. The 09 test set is almost completely distinct from the 2013 and 2016 ones. They had a slightly different approach to constructing it. But this is also partly one of the reasons that I actually just, for most of the analysis, looked at what happened when you combine all three into a big test set.
Yeah. I think we have to move on. Thanks a lot for the discussion. So our next... thanks again.